Okay, today we're going to talk about cell transportation, and some way that cells get things in and out of the cell are through osmosis, diffusion, and then other forms such as facilitated diffusion and even active transport. And again, these are the ways that cells can obtain the nutrients they need to maintain homeostasis and also to obtain and use energy. So pretty important concepts here. So let's go ahead and look at them. First off, let's talk about diffusion. Diffusion is just a random movement of particles. And anytime you have a gas or a liquid, you have molecules in motion. There's a lot of kinetic energy there. If we're talking about diffusion across the membrane, as long as we have molecules that can pass through that membrane, and remember we said molecules such as water, carbon dioxide, and oxygen can pass across the membrane, well, what'll happen if they're moving? Well, they're randomly going to be running around here, moving in all directions, and some of them are going to start moving over here, and we're going to see over time that we see this diffusion across that membrane. Now, how long will this diffusion occur? Well, diffusion never stops. Pardon my dog. Diffusion never stops, but it does reach what's called equilibrium. So what happened here is things haven't stopped. These guys are still moving over here. These guys are still moving over here. But we now have about the same concentration of water or oxygen on both sides of the membrane, and we've reached this state called equilibrium. What happens if we have two different solutes, two different substances that can move through the membrane? Again, well, diffusion, random movement, these guys are going to be moving, and a couple of them are going to move over here. These guys random move, and some of them move over here. So over time, we start to see diffusion occur as the purple ones are moving inside the cell, the yellow ones are moving outside the cell. And again, how long does this occur? Well, diffusion doesn't stop, but the net flow across the membrane is going to stop when we get an equilibrium, a state of equilibrium. In other words, about the same concentration on both sides of the membrane. Again, movement across the membrane is still occurring, but no net change. Okay, so now let's look at water, and osmosis is the diffusion of water across the membrane, and I'll talk a minute how that occurs. But because water can move across the membrane and sugars and salts and other things cannot pass through the membrane, that means that sometimes water can change cells in ways that can be good or bad. So let's talk about, let's say we put a cell in a pure water solution. We have a greater concentration of water outside the cell, less concentration inside the cell, and again, water can pass through the membrane. So what's going to happen? Water is going to flow in at a greater rate than it flows out. The cell is going to swell up, and the cell is going to eventually lyse and rupture. If it's a plant cell, it'll flow in, but because the cell membrane hits that cell wall, the cell wall will eventually increase in pressure and create what's called turgor pressure, and the cell will not rupture, but it will fill up and be what's called turgid, meaning it's got high pressure. Our cells, our kidneys help maintain our body in what's called an isotonic solution. Iso means the same, and again, tonic is solution. So an isotonic solution, we have the same concentration of water inside as outside of our cells. So water is moving in at the same rate water is moving out, and we get no net change. And in a plant cell, same thing occurs. Water in, water out. Flaccid just means it's not pressurized, but it's not shriveled up either. It's in sort of this isotonic condition where diffusion of water is occurring, no net flow though. Okay. Now let's look at putting a cell in a hypertonic solution. Now we have less water outside the cell than inside the cell. And what's going to happen here is we're going to have more water diffusing out of the cell than in the cell, right? And the cell is going to shrivel. And this happens with animal cells or plant cells. We'll look at plant cells in the lab. We'll look at some red onion cells and see this occur in them. But again, this is going to mean the cell is going to shrivel up. The membrane is going to shrink because it's losing water. Why is it losing water? Because it's going from higher concentration inside to lower concentration, that salty solution, outside the cell. So in the lab, we're going to look at some red onion cells, and you can put them in fresh water, and they're going to look like this. Now, you can't see the cell membrane, but if you could, it would be right up there against the cell wall. Why is it right up there? Because that high concentration of water outside the cell is causing water to want to go in, and it's going in until that membrane reaches the cell wall, and then, of course, the cell wall provides pressure that prevents it from lysing. So that's what red onion cells look like in fresh water, what we call a hypotonic solution. Then we're going to take your onion cells, we're going to put them in salt water. And when we do that, all of a sudden they look like this. Well, notice the cell walls have not changed. Cell walls are not selectively permeable, right? There's the cell. The membrane has. The membrane has shriveled up, okay? What's happened? Well, we've had more water go out of the cell than in the cell, and the net flow, osmosis, means that the cell has shriveled. Movement of water out of the cell has caused it to decrease in size, okay? So let's talk about some of the ways that things can move into and out of cells. We talked about how oxygen, carbon dioxide, non-charged small molecules can pass through the membrane, and they'll always pass from high concentration to low concentration, like is going on right here, and that's called diffusion, diffusion across the membrane. Okay, a specific type of diffusion where proteins allow things to pass through that usually can't pass through is called facilitated diffusion. So remember I said water can pass across the membrane and salts and sugars can't. Well, water actually can't pass through. Water is polar, and these non-polar fatty acids don't like water. But fortunately, our cell membranes have these channels in them called aquaporons that open up and allow movement of water through the cell membrane. So an example of a facilitated diffusion is movement of water through these proteins called aquaporons. So that's a second type of movement through, again, diffusion, still going from high concentration to lower concentration, but it's aided by proteins, so facilitated diffusion. Then the third type of movement across the cell, notice we have a lower concentration of particles out here than in here. And to move something from low concentration to high concentration, that's going against the way it wants to diffuse. The random movement, these guys would want to be moving that way, that way, that way, not inside. So to do that requires energy, and the energy molecule is ATP. So we have to put that energy in there and then sort of pump them across the membrane. And that takes energy in the form of ATP, and that's called active transport. I'm sorry, the first two processes are passive transport. The third, using energy, is active transport. Okay, so those are three types of movement across cells that we're going to be studying this week. And then when we talk about facilitated diffusion, there's two ways the proteins can move things across. Number one, we talked about those aquaporons that allow water to just move right through by opening up a channel. Those are called channel proteins. We can also have carrier proteins where they'll bind to a specific particle and move it through. And we have examples of these in our cells in our lungs, where we have too many chloride cells in our lungs, and they'll actually carrier proteins will bind to them and move them out of our cells and allow our cells to maintain the proper salt concentration. If we don't have those proteins, you have a disease called cystic fibrosis, where you can't move the excess chloride ions out of our lung cells. But again, these are examples of facilitated diffusion, proteins allowing things to diffuse from high concentration to low concentration. Okay? Then active transport. This diagram is showing active transport, and you can see that active transport involves the protein binding to a specific particle and then moving it from lower concentration over here to higher concentration over here. And again, that requires energy, and energy is in the form of this molecule ATP. And ATP actually sticks a high energy phosphate on here and gives it some energy. Then it can use that energy to pump that ion from low concentration to high concentration. But again, that process is called active transport. So these are ways proteins can move things into and out of cells. But sometimes we have molecules or particles that are so big that a protein can't actually get them in and out, right? So we have like white blood cells taking in bacteria. Bacterial cells are too big for a protein to kind of squeeze it in or create a channel. So we can actually have the entire membrane wrap around a particle. So maybe this is a really large food, like a starch molecule, or this is a bacterium in a white blood cell. And if we wrap around it and take it in, that process is called endocytosis. Endo means into, cytosis, cell. So the membrane can wrap around that particle and move it into the cell. And let's say it's a bacteria and we want to get rid of it, or a food molecule. We can fuse it with a lysosome. Remember, lysosomes have these digestive enzymes, and they'll break down the food particles. The food particles can then be taken into the cell. And let's say we just have some waste products that we don't want in the cell. Well, we can keep those within this container, maybe this vacuole, and we can fuse that vacuole again with the cell membrane. And again, since all membranes are interchangeable, when it fuses, then those particles can leave the cell without ever having passed through the membrane, and that process, when it exits the cell, is called exocytosis. So the opposite of endocytosis is exocytosis. So these are two ways that the membrane can bring things in and send things out without ever having to actually have things pass through the membrane themselves. So these are some ways that cells can move things in and out and again help maintain homeostasis and obtain all the nutrients that they need.